Yeah, ladies and gentlemen, welcome. Another week of biomedical microsystems. Um, we have finished the first round of fixing dates for exams, and I promise to come back with a date when I know something. This date should be circulated via the HIS-in1 system soon. You should check that you register for the exam or deregister if you don't want to participate. You know the usual things that you already know. And the date that I found in the system was August 31st at 2 p.m. So 14:00, 90 minutes, written exam. Rooms will be distributed shortly before, and you need a pen and a ruler. That's all. Good. So, here we go. Today, I like to share some thoughts with you about intracortical brain interfaces. So last time we had vision prosthesis. That's all a bit, let's say, cutting edge, really research. But the reason to share that with you is A, to show you cool things that are around, and B, to give you an idea how small microsystems can be used as tools, as probes to interface with the nervous system and to find out the challenges that we face if we do something like that. This lecture is mainly based on research things. Some of them are getting closer to medical applications, but this is a long way. So, and long means more than 15 years. So on average, if you are really educated in that field, if you have a good team, if you have 3-digit million US dollars, and you know what you want to do, then it takes you on average 12 and a half years from an idea to a medical device that is approved. If you're lacking either knowledge or money, or you have some bad luck with decisions, it can take even longer if you go towards implants. And that is probably, and people sometimes think I'm faster. No, I can put more money together, more persons, whatever. You can probably save 2 or 3 years, but it's a really long process for implants. If you do app on your smartphone, that can run in 2 years or something like that, but this is then a different story. And therefore, a lot of things here have been invented when, let's say, society and the rest of the technology was not yet mature enough to bring it into product. I think we're now at a different level, and therefore it could be that at least in your lifetime you might see 1 or 2 applications of those things in humans in general applications. From the technological side, this lecture is about silicon, mainly. At the end we go a little bit to polymers, while last week was mainly on polymers with some bits and pieces from silicon. This week is just the other way around. To check what can we do when we use microelectronics technologies and make something out of that to interface with the brain. So that's the storyline for today. So how did I structure that lecture? So I like to start with some fundamentals. And then go to the, let's say, dinosaurs of brain interfaces made out of silicon. That's the Utah array and the Michigan probes. And then I like to share some other thoughts with different stuff. Neuroprobes is something that has been co-invented here in Freiburg. and some thoughts about polymer-based systems at the end. I intentionally left out all those movies from Elon Musk. I have to admit, I'm really impressed how much he did over 2 or 3 years, and this is a lot of money, a lot of really high-end scientists and engineers. Put everything together, lock them in a building, give them enough food and drinks and video games and whatever, and let them work. It's impressive what he did. However, I do not like the marketing he does with the Neuralink. So that's a bit of overselling, and the things he did is the best from everywhere. So it's not that much where I would say this is really new, but it's really impressive how he managed to bring all those things together and drive the persons towards a common goal. And therefore I thought it's probably more informative for you if we jump back a little bit to the very beginning of those probes to see what were the ideas, how did they evolve, things like that. And we have to start with a question, and that is the question, why do people like to interface the brain? And that is not because they want to type faster in Twitter. That was not the origin of that. That's probably one of those driving forces today. But in the neurosciences, it's still to gain fundamental knowledge. We still do not know how the brain really works. I mean, you could say now, yeah, but I know if I move my little finger, there's a piece of the brain which codes that. And I completely agree with that. But if I would ask you, how do you know how the strawberry cake of your grandmother smelled? Yeah, this is much more compelling. And when you find press releases like we are able to read your thoughts, you should go to a dictionary and look up what does thought mean, right? We are probably able to record signals. We can assign signals to certain actions or to certain sensations, but I would say we are far apart from reading thoughts in their whole complexity that we have. And therefore we still need fundamental knowledge still. From the engineering side, sometimes we don't care about the knowledge. We have an idea, like there's vision prosthesis, and we say, hey, come on, no matter if we exactly know how it works or not, wouldn't it be cool to restore some sensations to improve quality of life of patients? Not saying that we fully restore that sense, that we have something that is probably a bit better than a cane or a stick or something like that. And we like to do diagnosis in some cases. We say, well, if you are from the outside, it's like watching a soccer game from the outside of the stadium and try to find out by all the screaming and shouting that you hear what's going on inside and how the situation is. And to use, as last point, those signals as command interfaces to drive assistive devices. And that is what you find in press releases as mind reading. Yeah? And I think we should go through those things. If, well, an engineering view of the brain, so we have a hierarchical organization, we have two hemispheres, and we know the basic unit is the neuron. And the question is, if we see all those different parts, how should we interface that? What is a good way to go? And if you ask two persons, you will get 3 answers. Even if we just focus on the cortex, with its cells and the fibers and its anatomical organization, it's not a black and white thing. You know, that it's very complex. There are processes going on that are amazing. You know, if I I have learned my English when I was 12, 13, 14, whatever, and older. If I would get a brainstem, or I would get a stroke in my brain, it could happen that I'm no longer able to talk in English, or that I lose my German and I'm able to talk in English, or my rudimentary French. If you grow up with two languages, that allocation of the languages is really intermingled like a salt and pepper pattern. And if you get a brainstem stroke, you cannot lose just one language, but you lose parts of communication or things like that. Blind persons who read Braille, for example, there's dots on the surface, you find activation of their visual cortex when they read that. And this is far beyond the black and white. You know, you would say, well, if I see something, I take my visual cortex. If I touch something, I take my somatosensory one. That there seems to be reorganization, learning, interconnections. It's not really like those ideas of electricians that you wire cables. That's part of the story, but that's not all. It's much more complex. And therefore, it's a good first educated guess if you want to do something with a visual system to go, for example, in the primary visual cortex. But it might not be the only truth. You know, if it's about hearing and understanding, for example, you have areas like the primary auditory cortex where the incoming information is processed. And then you have higher orders of processing areas where you can, for example, decode a pitch that goes up, no matter where the tone starts, right? So it's like melody decoding in certain things, or there are in Broca and Wernicke, those two areas of speech decoding and generation. There are brain strokes where you're no longer able to memorize certain words, but you still can assign other words of that class to that. So you're not able to find the word peach, but you find apricot because that's for you probably the nearest neighbor of peach. Yeah, that's really amazing. And therefore, we should on one hand be very pragmatic, on the other hand understand that this organ is very complex and that we're probably not able to decipher all of its secrets by 10 contact sites or something like that. And I'm pretty sure the answer could not be to have 10 trillions of contact sites that we poke into the brain. Even if they're very small, that's a very simple calculation. You know, if you have something that is very small and you multiply it by 3 trillion, that becomes quite large, right? And therefore I don't believe in that 10 million recording sites paradigms. We have that motor cortex arrangement, which is true, but there's more behind that, so we cannot really take it one-to-one. But the idea is that certain parts of the motor cortex code for certain parts of the body, and you can strengthen them. For example, by playing the violin, the hand area will get larger a bit. There are also observation if you have an amputation, for example, and there is no input that neighboring areas might overtake that. But this is high-level description. Some details are still unknown there, and we should keep that in mind when it comes to interfacing with the brain. We have about 10 to the 9 nerve cells in the brain, and one approach could be to try to implant 10 to the 9 contact sites that you record each and every nerve cell in the brain simultaneously. At best, at a higher recording frequency than the neurons fire, and then I leave it to you to calculate how much memory you need if you would record, let's say, with 10 kilohertz or 20 kilohertz, 10 to the 9 neurons with a resolution of 12-bit. And then you can ask yourself, who should analyze those data? Yeah? And the answer should not be, I'll take a convolutional neural network. That's probably one of the answers, but the question is this sufficient to get really the knowledge out of it? So we have chemical information, we have action potentials, and we have those postsynaptic potentials. That means if cells communicate with each other, it's not yes or no, but they have interconnections and they sum up spatiotemporally that information. And this is all we should keep in mind. And as I said, 10 to the 9 neurons, should we really record all of them? Is it probably sufficient to have a kind of network recording? If you say, oh, this is where I move my little finger, and if I get a signal there, isn't that good enough? Or, as things started, that people say, well, I don't want to drill a hole into somebody's skull just for curiosity reasons. I would prefer to place something on the surface of the skin, well knowing that we have the skin and the skull and whatever in between, and this acts like a spatiotemporal filter, and we lose some information there. But the question always is, what do I want to get out? So what is my question? And then to select the adequate tool to answer those questions. That is the question here on the level of neurons or populations. And that is the question of time. So what do I like to monitor? You know, if I need a legally binding signal that the person is dead, yeah, that's the EEG. EEG is one. If you have no longer your head, if you're fouling and smelling, or if you have a flat line. These are the three legal signs for being dead. I mean, you don't have to resolve that with 100 kilohertz. You know, if you're dead, you're dead. So you have time to measure that. The question is, do I really always need that millisecond resolution? Yeah? I have no good answer for you with respect to neural signals, but I know if I get fever, that doesn't change in the microsecond range. So I do not need to measure my fever 100 times in a second. So that's probably good every hour, 3 times an hour, something like that. And that gives you a lot more room to transmit data, save power, save dissipation energy in your controller if you are allowed to be slow. And the question, what is the spatial resolution? And you see a lot of abbreviations here. fMRI is magnetic resonance imaging, so it's non-invasive but slow with a medium resolution. We have EEG, which is a bit faster, but the resolution is also not that good. We have magnetic measurements, MEG, magnetoencephalograms, so you don't take the electrical signals, but the magnetic counterparts by the electromechanical coupling. You can open the skull and put arrays on the surface of the brain, where you get rid of the, let's say, insulation properties of skull and skin. You can record local field potentials. You can jump into optical imaging techniques, either on blood oxygen saturation, on fluorescence imaging, on calcium, on optogenetics. So there's a broad range, and you can finally end up with multi and single unit activity, which is fast and fine microns resolution. So, and that is still an ongoing discussion, and some 10 years ago people said, well, if you really, and it was about movement decoding, so it was not about thoughts and mind reading, but really straightforward movement decoding. So if I move my hand in space, how do we decode that? And the hypothesis was there might be neurons which code for certain directions. So if I move my arm forward, there is a forward neuron. If I move my arm backward, there's a kind of backward neuron. And to get redundancy, we do not have only one, but some numbers there. And if we now find those neurons and record them and have a calibration procedure that we know, oh, this is forward, this is backward, in a kind of, I record, move my arm in known directions, I can then predict the arm movement afterwards by recording of the neurons and putting them together in a time series, which is called a trajectory in space. So time series over space series, and by recording those neurons I know where I want to place my arm. That was one of the iconic discoveries here. And the challenge is to find the neuron. If I stole that from colleagues from Michigan University in Ann Arbor, so if we would have a column 1 millimeter by 1 millimeter by 2 millimeters, that would be about 60,000 neurons and 1.4 to the 9 synapses. And on average, we get, if we have probes without electronics, about 16 contact sites in. Or we have now the new, like the neuropixels, which are that shallow as one of those shanks, and they have 1024 contact sites on one of those shanks. Really cool. A lot of electronics in the background, and you can switch with a resolution of about 20 microns between the contacts. But you still cover then one thing here. And there's now 2 years ago, a US funding agency from the military, the DARPA, announced the 1 million channel challenge. So, make your thoughts and give us a short description. How would you like to solve that problem to record 1 million signals in the brain? Really cool approaches, but and you have to think about that. Let's make a thought experiment. Take wires, and a wire is half a micron thick to be close to the nanospace, and you have 1 micron distance to the next wire, and a million is 10 to the 6. So that means 1,000 by 1,000 is a million. So you would have an array with 1 million contact sites made of imaginary wires, and you would put that in the brain, and that would cover 2 millimeters by 2 millimeters. That means it's very cool for that spot, but what if your spot is somewhere else? Then you would have to take another 1 million channel array and another one. And if you want to have the coding between, let's say, sensory feedback, motor commands, vision to investigate hierarchical control between tactile, proprioceptive, and visual feedback, then you would end up with about 100 to 1,000 of those 1 million channel devices. And that sounds completely cool, but the question is, how can you really do that? Is this a good approach, and do you get that information out of that you would need? And I cannot give you an answer. I just want like you to think about that. Yeah, if people come with incredibly large numbers of things. And again, 10 kilohertz sampling frequency, 12-bit resolution. That's an unimaginary size of hard disks that you have to fill in first, and then to find out which kind of data do I need, what is noise, what's really the desired signal that I get. And if you do not know what you're looking for or what your hypothesis is, then it's really hard to take artificial intelligence signal processing, because if something comes out, you don't know, if you don't ask the right question, if the answer is somehow linked to your question, right? This is one of the main challenges there. This was here with that directional coding from Apostolos Georgopoulos in 1982, his iconic publication, now cited thousands of times, that he found out there's a directional neuron there. And if I move my arm in those 8 directions, I get firing neurons, and you have different recording channels, and whenever an action potential occurs, you draw a dot. And the more dots you have, the more active it is. And then you find out, okay, in this direction, these bodies are really good, and others are good in other directions. And then there's a certain display that you combine the number of firing neurons with the length of a vector and the direction in which they are. So is this only my one, or are there neighboring that fire here in other vectors? And then you superimpose them and get a kind of population vector that points in a certain direction. And then you can use that for decoding. We still do not know how to record, but you get now an idea what the idea behind that is. Yeah?
Can you return to the previous slide? Okay. If I have more dots on the left side, or if I do my arm like this more than the right side, how does that affect my body? The reflection on having more signals?
No, it just tells you if you move your arm to one side,
Okay.
you find it out because of that pattern during recording.
Mm-hmm.
It's not good or bad, it's just, let's say, a mirroring of the movement to a certain nerve activity pattern.
Okay.
And that means if I move here to 180 degrees, those population is firing, while if you move to 0 degree, it does not fire. And by differentiating of those two patterns, when you just record, you know, oh, this is my intention to go to the left. And if you see the other pattern, you know, oh, my intention is to move the arm to the right. So that's very important. It's just an observation. It's not an assessment if it's good or bad or doing something. It's just I'll do something and I'll find out this is the pattern that is assigned to that movement. And then you can use a classifier or whatever and sort that out in different movement directions. So, and there were, I'm now cheating a little bit. The second approach that I'll go to show is the one that was first in time. But I decided to do it that way around because this is kind of more intuitive for me. So what do we have? We have two approaches, and I've chosen those two approaches because they illustrate two ways of engineering those interfaces. And again, there's no good or bad. There are good reasons to use the Utah array, and there are as many good reasons to use the other array. And then you have to find out which is my application, what fits better to that particular application. So the Utah array is a bed of nails, period. A bed of nails made out of a single block of silicon. And that means the length of the needles is associated to the block of silicon. And you do not yet, well, you can buy it for gold or something like that, but you have standard thicknesses of certain materials, and this is the limitation here. And we have a length of the needles, that means the thickness of the block, of close to 2 millimeters. What do we see? We see the needles, they are kind of sharp at the tip, and the tip has a different color. And that, I'll tell you some secrets now before on that slide. This is here, that very whitish stuff is the recording site. So this part of the shank that is conductive to record electrical signals, and the rest is insulated. And this is important because if it would not be insulated, you could not selectively record from the tip. If you have a whole wire of 2 millimeters that is conductive, you record everything over that. And you could either say very positively that is a kind of integrator that integrates all signals over the whole area, or you could be a little bit mean and say, well, it short circuits everything over that length. Two sides of a metal, either if I say it's an integrator or if it integrates everything that you do not see the details any longer. Two questions. A, how do we make that? B, how are we able to contact those single things there? And C, how do we get that into a brain? Right? 4 by 4 square millimeters. So brain, very squishy, soft tissue. If I have something like that and put it on the surface, it might dimple. And you might know that from a balloon. If you try to poke a needle into a balloon, it explodes in most cases. If you put a piece of tape over that it's not able to explode, then you can poke it in. And here I can tell you that in advance they did a lot of experiments how to bring it in, and they came out with an air gun. So you put it on the surface, you take an air gun with a stencil, and the reason for that is if you look on basic physics, you find the impulse, yeah, delta F over delta T, and if you are very fast, that soft tissue, the inertia is too high to dimple in that short period of time. That means it penetrates because of that high impulse that we have, that short time over there. But the question, how do we make that? And they took a piece of silicon called a wafer, and now the task is to make a conductor between the back part and the front part without short-circuiting adjacent areas. And what has been done in those days, and this goes back in the late 1980s, early 1990s, to put aluminum on the back side and then have a strong temperature gradient over front side and back side. And if you look into material science, we have an eutectic alloy between silicon and aluminum. That means the melting point of that alloy is lower than the melting point of aluminum and lower as the melting point of silicon. That's the definition of an eutectic. And by that, you get the silicon aluminum eutectic, and if you choose your gradient between front side and back side strong enough, which is not that easy, then this migrates through. The wafer was an N-type, that means negative conducting, and this is now positively conducting. And the plus means we have more positive charge carriers than there by number, as we have negative in the wafer. Remember basic physics, electrical engineering. If you have semiconductors and you have a plus and you have a minus, they form an interface layer. And this interface layer, PN diode, the very tiny interface is free of charge carriers. That would mean if there are no charge carriers, there is no conduction, and this could act as an insulator. Thereby, we have conductive pathways insulating islands around. Nevertheless, this is still a slice of silicon, no bed of nails. Next step is to take a saw and to cut a chessboard, checkerboard out of that. That's good, but it's still square shaped. And if we look here on those, this is now freestanding. We don't care at the moment about insulation, but we care about the insulation of adjacent posts here. And since we have that PN transition or junction, we have here in between those two needles a transition which is non-conducting here. So kind of insulation. I can tell you later on they completely quit that and put glass in between, which insulates much better as semiconductor PN transitions junctions do. And then everything is put into very aggressive etch. Hydrofluoric acid, HF, is the most aggressive etching agent you can imagine, and nitric acid. And by that you round up the square-shaped posts. You know, those square-shaped posts have corners, and the more cornerish something is, the more aggressive are etchants or electrical fields, so we round that up. And if we do some tricks with inclination and moving things, we can even etch them thinner at the tip. For that, we have semiconductor material needles standing around, having neither a metal interface nor an insulation. So, by shadow mask, and shadow mask means you take an aluminum foil, you poke that stuff through, and then you put it into a metal deposition machine, and then metal is deposited, and then you rip off the aluminum foil at the end, and you have only, if you're lucky and you did it all well, you have that metal on the tips. That means we have now defined tip for electrical recording, and the question is, how do we get insulation? And this is that has been tried out with different materials. Could be polyimide, could be Parylene C. I think the current insulation is Parylene C. So deposit Parylene C everywhere and take a laser and shoot off, or plasma and shoot off the metal from the material from the tips. Then the metal is underneath with a metal tip and an electrically insulating shaft. Backside contacts have made by ultrasound wire bonding, so piece by piece, putting a wire, vibration, heat, and pulling that wire along, and then a person who's not well paid with small hands has to solder the other side. And then you put something insulating on top, like silicone, rubber, polyurethane, whatever. And those wires are still the main breakage point of those devices. Just by movement, by handling, wires are always the bad boy here. We can model that with the insulation, with the openings, and all that stuff, and then we can implant that. And we still now, 21st century, this is from 2006, we are now 2022. The only thing that has changed beyond reliability, so a lot of tiny little things that no one sees, that those arrays are now more stable and are able to last for about 5 years. That's the longest they have been implanted. They still come with that really ugly percutaneous post. So you go into a hardware shop, no you don't, you order probably from a medical device supplier, some bone screws, and then you drill bone screws through those holes into the skull to fix that thing. And then the skin, as shown here, is placed around. And if you're good and undergo sufficient hygiene, it does not, there's no inflammation. On the other hand, this is really the tricky thing, you know? You have that on the skull, that is ugly, but it doesn't matter that much. But what matters is the cable that goes through the skull into the brain that might act as an entry path for germs. Over the weeks and months, this is closed, so bone grows around that, but for the first weeks, that's really serious. And you see here those tiny little cables going to the array, and probably you see some other wires going on here. That's ground and counter electrode. So if you have an amplifier, you record, and then you need the second pole, let's say if you do monopolar recording as a kind of reference or counter electrode, and then you quite often have a third one that you get a kind of middle potential for your operational amplifiers. This is the most famous implantation, and that is the reason why I brought that one. I have newer videos from newer publications, but this is really the most famous one here. I'll like to show you the patient in a second. There's no blood, no open skull, nothing. It's the performance of the patient here. That was a C3 patient. C3 means cervical level of the spine. C3 is that high that you can no longer breathe by your own. That means this patient has a tracheostomy for inflation and has to learn how to use his motor signals, that is in the motor cortex. That was the reason why I showed you that movement decoding at the beginning. By movement decoding to interact with the computer screen. And he got very famous for that. Let's see, yesterday evening it still worked. Let's see if it works that way. Aha. So that doesn't work as expected. That's a pity. Let's see if it works in the other scheme here. If not, I'll show you the video on one of the next lectures. No, for some stupid reasons my computer did not copy the movie. Apologies. So what you would see would be where this guy is able to move the cursor on the screen and can, this is an old style computer graphical user interface, so switch on the television, switch between channels of the television, increase the sound and decrease that, and then scrabble something on the screen. What is still the same, well, nowadays it would be still the same with a ventilation machine. There is still the option with that wired preamplifier system that you have there. Now since 3 years they have a wireless system. That means you still have that post, but you can screw on a kind of Wi-Fi system there that you can communicate between that with your central computer for cursor control. There have been also investigations to have a robotic arm control. People are now there are about 40 persons who have been implanted It's still not a product, still a research device. The US has that option to have an IDE approval. IDE stands for Investigational Device Exemption. That means you have to prove that your thing is safe, but it's not yet a medical product, but it's an investigational device which proves safety, and then medical groups can apply with a research proposal to investigate things. There are now about 40 persons having that. They're all closely linked with a company who distributes them, and they really participate in research. They're keen on helping out to contribute to that research that future generations can probably benefit from that. There have been approaches. Your question would be, why isn't that wireless, right? It's so obvious that it should be wireless, and there has been a lot of research on that, starting in around the 2000s. Why not putting here a wireless system on the back part? You have 2 millimeters or 3 or 4 by 4, so there's some space, right? And there have been developments there to have coils, so different frequencies that in-link and outlink. So outlink would be the recording of the signals, and the in-link would be, let's say, the adjustment of the amplifiers or the selection of the amplifiers. You don't need 100. You probably record from 4 to 10 channels, but those channels might vary, so you must be able to switch in between them, to lock them, to have the right amplification, stuff like that. Therefore, 2 coils. They found out there should be a ferrite plate towards the electronics. Otherwise, right-hand rule, you have a coil, the current goes through, you have a magnetic field, that magnetic field goes into your electronics, and when it changes, change of magnetic flux over time, dB over dt causes an induced voltage, and that induced voltage shoots off your amplifiers. Something you don't want to have. So never forget the coil interaction with your electronics. Therefore, it's that ferrite plate that decouples the magnetic field from receiving and transmitting from the underlying microelectronics. And you always need capacitors for storage of energy to drive something, and spaces that it does not wiggle around. And the coils, and that manufacturing process was a nightmare. You can imagine, you have that bed of nails that is, brittle and delicate, and then you have to assemble that. And you have to coat that, because that also goes into the body and is wet and should not get destroyed. And the other idea was, why not putting a ceramic package around that? And at the very end it came out that this is all good for research, but it takes ages, it costs too much money, and the yield, that means how many things do really work when I'm at the end of the manufacturing line, was not good enough. And then they said, well, we don't have, our research project is finished, but if we transfer that to a company, nobody would pay the price. That was about 2006. And I know that there are things going on in the background, but the company then said it's more important to make those Utah arrays, those bed of nails, work in patience. I would not say they're happy with the percutaneous plug, but they don't complain too much, and therefore that was further proceeded. So that's, let's say, 50% of the world works with a Utah array. This is particularly good for chronic implantations in both humans and non-human primates. If you want to investigate certain questions where you need the interaction between different brain areas. So and up to 4 or 6 of those arrays can be implanted in non-human primates. You have those head posts, and then you can undergo fundamental experiments. But all those contact points that you have are in the same distance. That means probably you're accessing layer 3 or 4, and you're accessing layer 3 or 4 everywhere. If you want to investigate what is going on between layer 1 and layer 6, these probes are not the correct ones. And this is where the Michigan electrodes come into play. And the first Michigan probe ever had 4 contact sites and was done at Stanford University in 1969. And those 3 guys were Angel, Starr, and Weiss. Iconic names. They did that once. Ken Weiss finished his PhD thesis and then went from Stanford to Michigan, and then it took him another 3-4 years to replicate everything again. So he's one of the fathers of the Michigan probe, and I was lucky enough to get him to know. I think he's now 86, 87, 88, something like that. He was still, some years ago, several times here in Freiburg on workshops and stuff like that. And what you see here is that this is called an in-plane manufacturing. Yeah? While the meter is when you have the wafer and the Utah array stack out of plane, wafer out of plane, we have here an in-plane manufacturing. We have the wafer and everything is made like cream cake on top of that. So what do we need? We need a substrate, so we can handle that. We need some contact sites, we need insulation, and we need interconnects. That means horizontal wires going from the contact site to a contact path. If you want to go out of plane, you have to get them apart from the wafer, have to turn them by 90 degrees and assemble them in a kind of platform. Or you don't assemble them, you just put them in a holder and say, I want to investigate certain interactions in the brain acutely, then you can do it during the procedure of investigation. Or you can also do that chronically, then you have to fix them somehow in a head chamber or some dental cement or something like that. But the idea was, can we put a lot of electronics on there, that we have more than 1,000 of probes, can we switch in between them with multiplexes, can we store something, whatever. So whatever you can imagine, and there's an iconic paper in 2004 from Ken Weiss and all the guys and girls who worked in his group on what could we do if we have endless amount of money and endless time. So how to bring that together. And you see, this is 2004. I told you he did it for the first time, 1969. That's 35 years to that, and we're still not at the point where this is an approved device. And the reason for that is that it has been mainly done for neuroscientific fundamental investigations. The purpose was never to make a medical device out of that. The company has been set up, Neuronexus, and they are commercializing those probes worldwide for neuroscience groups. Their process, and this is famous, their process, because they thinned the electrodes down from thick 500 micron wafers to 30 microns, which is less than a tenth of that. You see the substrate, this is the back part, an electrode site, and the top layers here in the scanning electron micrograph are insulation materials. The beauty is it's very thin at the beginning, so if you have something that's very thin and very sharp, it penetrates easily, so beautiful. You have that transition, that means it does not create that much damage, it's not like massive post. you have to put in. But imagine if you have an asymmetric thin piece of something and you push, then fundamental mechanics, imagine where the neutral fiber would be. And it's not in the middle. That means if you push and you cannot look and you have transitions in Young's modulus, so on the stiffness of the tissue, it could be that that thing bends to one side or the other because of that asymmetry. And this is indeed a challenge if you get too long with those probes, that they tend to turn in one direction. This is the process here, and I have to introduce a definition of passive and active here in that field of neural probes. And passive always means that we have no electronics. Passive means we have a substrate, we have insulation materials, we have metal lines for interconnects, we have contact sites, but we have no multiplexes, amplifiers, transmission electronics. If we have that, then those probes are called active probes. Here, this shape is a combination of diffusion. So upper left, we diffuse something in, and it forms more or less an isotropic shape. Since we have it wider, we don't get a semicircle, but we stop before we get that, so we have a kind of roundish thing. And this is very highly positively doped. And if we put that into EDP, and the long name is above there, if we put that into EDP, EDP etches everything but highly positively doped silicon. That means that doping front forms a natural etch stop. So if we put it in, and it does not etch silicon oxide and silicon nitride, so our insulation stacks here, and it does not etch gold or platinum or iridium oxide, just silicon. That means all silicon has gone away, and the rest is positively doped silicon and the insulation material. As you see, this looks here really like a pastrami sandwich. A lot of slices here, one over the other. That is good, because the insulation layers are insulated from each other. And we do not care at all that this is conductive, because the conductive substrate is electrically insulated from the rest of the probe. Very amazing to me when I read it first is, if you thin silicon down to less than 30 microns, it changes its properties. Normally silicon as a single crystal is very brittle. So if you smash it on the table, it goes into bits and pieces. If it's thinner than 30 microns, it gets flexible. That means you can really bend it, you can even make a knot into such a probe or a cable. And that was the reason why the Michigan persons, I think it was Jamie Hetke in those days, she invented boron-doped silicon interconnects that are flexible. Over the years, it turned out that's really cool, but it's too expensive. You know, if you have a silicon wafer on which you could do a lot of microelectronic circuitry, and you just make passive cables out of that, it's a kind of wasting material. And therefore, they changed from silicon-based cables to polyimide-based cables. They're as flexible, even a bit more flexible, and much cheaper there. And that was in the late 1980s, early 1990s, if I remember well. And in parallel, they thought, well, we have silicon, we should take advantage of that. You know, if we take silicon, we definitely should integrate electronic circuitry in there. And that is shown here. The trick is you have to do it in 2 steps. First, to make your mind that you do everything that is kind of CMOS, so all the electronics part. And this is done in an electronics clean room, and they are really strict on having certain processes and not having others, because of optimizing the electronics performance. And that would mean you do all the electronics here, and I don't want to dig too much into detail what a clever design of CMOS electronics is and what is analog and digital. I think that would go far beyond the scope of that course. For those of you who are not that deeply involved into CMOS, CMOS is complementary metal oxide semiconductor, is the standard electronics manufacturing technologies over the last 50 years, 40 probably. So we make all those tiny little things that you have in all of your gadgets, all the electronics. But this is not good enough for our probes. So we can do here the formation of the substrate, and we can do all the electronics here, and then sometimes those things are changed into second clean room, and this is then called post-CMOS processing. That means now the focus is on stability in the human body, so oxides and nitrides that are used are more dense. For example, you are allowed to use gold and platinum, which you are not in a CMOS clean room for good reasons. So gold, for example, completely changes your electronics performance if it happens to go in there. So there are good reasons that they only use aluminum, but aluminum corrodes immediately. That will mean we take that and then you say, okay, now I have the electronics, I have to put gold over every open aluminum pack, for example, that it does not corrode. Or I have to put on a second insulation layer that withstands the salt environment in the body. Things like that. And at the end, you throw everything in an etching bath of EDP, and then you get the probes. They were very creative in combining them with also microfluidics. That means you have to make a channel into silicon. First, open a channel, and second, close the lid of the channel, otherwise it's not a channel, you know. And that was really demanding, and they played a lot of tricks here. For example, to have that chevron-like pattern of the surface layer, which allows the entry of etchant solution. Then you etch out that groove. And for the experts of you from the MEMS side, here the orientations, crystal orientations of the wafer that give us here the characteristic angle of wet etching if we use KOH, for example, and that chevron pattern. That's silicon. And if you do then, after that, a thermal oxidation of that, that means you grow oxide out, you grow silicon dioxide by adding oxygen and a bit of water vapor. And by that, you would change the surface and all the edges here from pure silicon into silicon dioxide. Silicon dioxide has 2 oxygens. That means it needs more space. And if it grows in all directions, those openings are closed. You, as an engineer, have to design it such that there is still an inner hollow area of the channel, because also all the other channel walls experience the same transfer into silicon dioxide. That's one of the challenges here. And to be honest, this is doable. This is material science, you know? That's difficult but doable. What is more pain in the neck is to put some fluidic channels on the end. Because this is something you have to do by hand. And then things become really difficult. You would love to have electrical connections there, and you need fluidic ones. And I think this is still one of the things why that never flew. Yeah, it doesn't fly because of all the connectors. And it's easier to put a pipette in for whatever you need, for dragging some fluid, for inserting some pharmaceutical agents, than having everything integrated. This is one of my favorite figures. We have here an amplification and multiplexing, 1 electrode per shank. I think this is here, right, 64 sites, so 8 channels per shank. And the probes are pretty small, and the electronics is pretty large. I have not my favorite here. My favorite is that we have that one combined with optics. And if you put it under a microscope, you have an addition of 16 or 32 microdiodes. And of course, you can then play music and make a bit of light organ in the background with that stuff. And then you zoom out, and your device is 3 millimeters by 2 millimeters, and the control electronics is like that, like a shoebox. And this is still one of our major challenges in all of those systems, and I believe one of those why they don't make it into clinical applications. That's beautiful for fundamental science. I think the idea of having hat-fixed humans over some days is hard to communicate, but you need really a fixed hat that you're able to record with that and to interact. So really high-end electronics, and they did a great job, and it's not an offense against them. It's a general challenge to make electronics that small that you can combine the beauty of those miniaturized systems with small electronics. And more clever concepts have been developed that you can shrink down the contact part of the electronics, even have electronics behind the electrodes on the shaft, and coming up with 200, 300 microns over a longer length, and then hand over that to the more sophisticated control electronics outside. Electronics always has to get packaged, and a lot of approaches have been introduced there, and this is just a warning. We'll dig into packaging a little bit deeper towards the end of the course to get an idea what does that mean, how can I make a watertight package around delicate electronics. Here there have been different approaches. First, to bring an insulator on top of the electronics. We don't short circuit the electronics, and then bring, for example, a metal layer on top of that, and the metal layer blocks all water vapor and all ions to interact with the electronics. And more details in lecture 12. Assembly, as I said, these are in-plane probes. If you want to have a bed of nails, you have to tilt them and make an out-of-plane assembly here. So with a platform, you have to align them. They should be really orthogonal. If they're somehow tilted, then it's hard to predict where they go, and it's harder to get them into the brain if they're in oblique angles here. And you need somehow electrical interconnects to have the electrodes and the electronics from the single modules connected to a cable. And this is how it looked like. Really beautiful 1024 channel array. And they had the cool idea of those wing flaps up there that are on each and every module, a little bit like springs. And you assemble them, and when the module is in, you come with a bonder, with a chisel, and by vibration and temperature, you get a conductive interconnection, so something in between welding and soldering between. But you also see this is a serial process. You cannot repair anything. Once that module is assembled, it's done. You know, if something breaks or gets loose, that's bad luck. And another thing, I discovered that many years later when we sat together here on a joint project on campus, is the insertion of the platform. The holes must be really tiny in which you put your probe, that you have good alignment. But if you have temperature changes, and that could be just 30, 40 degrees C for whatever, the thermal expansion is that large that it's really hard to get that stuff in. And that means that assembly turned out to be one of the showstoppers of the whole approach. Not only with us, also the Michigan guys decided this was a good demonstrator and we stick to single modules without assembling them into larger ones. Beginning 2000s, Freiburg came to the scene with the Neuroprobes in a huge European project, and the idea was, and there it was just curiosity driven, you know. We want to show that Europe is as good as the US, kind of competing things. In the meantime, we have to say, due to interrupted supply chains and problems of getting materials, we currently have to check out where to get certain gases because the supply companies told us, oh, you need gas, that's beautiful. Our containers are in Shanghai. Delivery date is Q4 2022, that means close to Christmas. So it's a nightmare, and it will not get better. Therefore, it's quite good to have certain hubs distributed all over the world. And we started with crazy ideas, and at the end we came to conclusions that others have also been done. So this is our approach. Patrick Ruther, Oliver Paul were the driving forces here. And Atlas Neuroengineering has been founded as company so 50% is here, 50% is in IMEC in Leuven in Belgium for those probes, and they are symmetric. So they're a bit thicker than the Michigan probes, but they are symmetric, and therefore they have advantages if they are longer. Then they have a more isotropic distribution of intrinsic stress and forces and do not tend to bend that much. And have those differences, so more than one electrode per shaft, also in plane, symmetric geometry. They started with a modular concept that was also as challenging as other person's modular concepts. And they also started with, let's say, more sophisticated microfluidic processes that you can combine electrical recording with injection of fluids, and the idea was to locally investigate, for example, drug interaction with the nervous system, to inject viruses for optogenetics. But it turned out that this is really a hard job. So just some slides here. Don't want to overdo it with the, this is probably as take-home message here. It's really challenging. Yeah, so a lot of steps here, and this is probably better. So we have those electrodes. They can be long, they can be thin, they can be shallow. They have several contacts, and we learned our lessons. It took us 4 years to communicate to our neuroscience partners that micromachining is not capable with that in-plane process to make electrodes on the side walls of the silicon. No, we can make them on the front side. We can cheat a little bit and glue two wafers together. Then we have them on the front side and on the back side. Well, we cannot have them on the side walls. And that was so obvious for us, and it took us at the end 4 years to communicate that. That means I'm now, because it was not that the others were not able to understand that, but we were not able to understand their questions. It was so obvious for us from the process engineering side, and so obvious from them to us, can we just turn that by 90 degrees, that we both did not understand each other's. And that means that's a reason why it's so important to sit together with different disciplines, to talk to each other, and to understand what the other really means. There are some other, let's say, urban myths in neuroscience and engineering, how things should be, and it takes quite a while to dig into that, to understand where they come from, and then to assess if they're still valid if you use a different technology. But this goes far too much into depth. So, this is with microfluidics, so the idea. This is a cross-section, and this would be the channel here to spit out the microfluidics. This yellow stuff would be recording side, and this is additional handling substrate that things don't break. And this is how it looked like. Modular assembly, but it turned out that only a few persons are really in need of such a modular assembly. And if they are in need, they're no longer interested if they see the price tag. That's also very important to know, that the price tag could be a showstopper for many applications. And it's a long way to convince persons that the outcome might be much better if they invest once. But it's sometimes really hard. And if you take a lot of probes in neuroscience are still handmade, so you take tungsten wires and twist them and glue them together with whatever glue you have, epoxy or something like that. And that costs nothing. And if I would show up and say, hey, I have something that is much better, it costs only 2,000 euros, dollars, or whatever, that's incredibly expensive, right? And if you then say, oh, well, I think this is a disposable, you can use it once, it's not a good business idea. So you have to prove that you can clean those probes again, that you can reuse them, that there's a clear indication that you see when it's done. So probably after 20 times they say, well, if you measure something like that, then you have to send them back, or stuff like that. So saying that, this is the marketing slide here. We're still working on probes, and we means still my group and Patrick Ruther's group. So if you're interested, I think we're happy to help you with those things. The fluidic stuff is a bit stopped. That was not really a business model. It was the idea to come together with pharmaceutical companies, and they were not that much interested. To close that story, I like to give you some hints, let's say, not comprehensively, but some ideas about flexible probes. That's my field of research, and I could talk for ages here, but I want to keep it short. The idea behind flexible probes is driven from chronic applications. If I want to do something acutely, I can stick something for, let's say, a minute or an hour into the brain, do my investigation, put it out, and draw my conclusions. And that is good, because if I have something stiff and I put it in, I know where it ends. That's good if I want to stereotactically target my nucleus or whatever. However, if I want to perform chronic recordings, everybody of us, no matter if we are mouse or human, we have to breathe. We have a heartbeat. And that is something that makes the brain pulsate. That means if I stick something in and my brain pulsates, that there is a relative movement. And everyone might know that if you have a splinter into the finger and it moves, it's fine, but if you touch something and you have a relative movement, it hurts. And that is the same in the brain. It does not hurt because we have no nociceptors there, no pain fibers, but that movement causes chronic inflammation. And chronic inflammation means active cells which want to eat up the foreign body, scarring around that, so the interface gets more and more scarry and more inflamed, nerve cells die, and at the very end we have no nerve cells that we like to record at that interface. So it doesn't make any sense. And the idea behind all developments is, can we make the interface as soft as the brain? And I still doubt if we can do that, because then an electrode should look like water. I have no idea how to interconnect with water and how to bring that in. So some people say we can use hydrogels, that's kind of stiff water, but still that's difficult. If you imagine a jellyfish, how to implant a jellyfish in the brain, and having cables coming out of that, right? So flexibility is there. It should be more flexible than silicon. It should not, if it moves, then it should move with the brain and not being stiff while the brain moves. That means the connector, the cables to the outside should be at least flexible, and there should be as little tethering forces on the whole stuff as imaginable. But it should get implanted. I mean, if you have a rope, you can pull on the rope, but you cannot push on the rope. Yeah, that means we need something to make it stiff to get it in, either by a shuttle or a coating that switches mechanical properties, or by something that dissolves. And this has been investigated over the last 20, 30 years. So one thing is either to fill or to coat probes with a material that gets stiff when it's dry, to implant it, and then it gets floppy and the probe floats around. The question always is, what does that material do with the target tissue? So if we take saccharose, so sugar, or PG, or whatever, polyethylene glycol, alcohol, what does it do? The second thing is to make probes a bit stiff. So, for example, to have stiff parts and have hinges to make them flexible. So if they're in, they can move a little bit. I would say after all those years, this is an engineering idea which combines the worst of two worlds. Yeah? So it's hard to implant and it's still stiff enough to make all the damage you can imagine. So I wouldn't do that, but I want to show it to you for completeness reasons. I had ideas, not reading my mechanics textbooks thoroughly enough, and I thought, well, if you have a sheet of flexible material like polyimide, it's really stiff in that direction and it's too floppy in the other one, so I don't get it in. Can't we make a three-dimensional forming like a U shape and put it in? Worked perfectly well, with the disadvantage that it's now stiff in all directions. Yeah? If you, research is accompanied with a lot of failures, you know? There's something like that, it's really floppy, but if I turn it like that, it's stiff in all directions. So was a nice idea, not the best outcome. And then we came at the very end to the most simple idea one could imagine. We just made a hole in the tip, took a tungsten rod, which is stiff, pushed that in and retracted the tungsten rod and everything. The question is does that damage the brain? And the answer is it depends. Depends who is doing that and how thick the rod is. If people take a very thick rod, say, oh, it's so tiny little probe, but rod like that into a mouse brain, then it makes a lot of damage. If you take a very tiny little one and you take care, then everything is fine. And everything is fine is the closing slide here. We did that in our groups here, Maria Asplund's group and my group. We joined forces and made, that was when NeuroPixel came out, and shortly afterwards Elon Musk's first press release came, and we said, oh, that's really a pity, because we're at least as small he is, and we could show something probably that is meaningful. So telling what we see. And so we joined forces, made those multi-layer electrodes with very tiny little contact sites, 15 micrometers by 15 micrometers, and had good luck that we were asked to do so by Jure Bozaki, who's one of the pioneers and leading guys, now being over 70 years old, from New York University. And he implanted those things, or his group implanted those probes, and we had stable activity over half a year with flexible probes without having any electronics around that, which is good. And the second good thing is that we could show that small enough is good enough. The question is, how small do you have to go? And we believe, and we have now data that support that 30 microns are small enough from the width. So that's still within the diffusion length where nerve cells feel happy. So of course, it's completely cool if you have probes that are 1 micron by 1 micron, or 2 microns by half a micron, or something like that. But the question is, who is able worldwide to implant them? I mean, if you have one lab worldwide which is able to use that, it's probably not that useful for the generation of knowledge and transfer in other labs. And so we were quite happy to show that proof of concept. That does not mean at all that we are at the end of the road, but it shows that this technology, even though it has not been neither graphene nor neuromorphic nor all those buzzwords in there, can still work for robust and reliable probes. And with that, I'm at the end, and like to summarize, here's the literature for those of you who want to try that out with our literature access options that we have at the University of Freiburg. I want to summarize here. I've shown you intracortical neural interfaces. One approach for brain-machine interfaces, so for controlling assistive devices, those intracortical ones. They're mainly silicon-based and have not yet entered clinical practice, even though they're human clinical trials. But it's nothing you can ask your general health insurance for. And those MEMS technologies, microelectromechanical systems, are really mandatory to get those integration densities. You cannot do that by hand. You need those technologies where you can photograph all those structures in that tiny little size and combine materials. The materials have to be carefully selected with respect to biostability requirements. Something I like to point out more in lecture 12 on packaging, where we see what can the body do with those materials. And if you want to have integrated circuitry and you want to do silicon circuits, then they should be CMOS compatible. There are now for more than 10 years organic approaches there, so organic electronics. We all know OLED TVs. Yeah, there's the forefront of organic electronics, but when it comes to implants, they're cool for research publications, but they're not yet that robust and reliable that it can really be transferred into clinic applications. So with that, I'm at the very, very end for today. Thank you very much. I wish you a nice and pleasant week.